Okay, so this is the third and last part of chapter 8. And in this part of the chapter, we're going to focus on the light-independent reactions of photosynthesis. And so this is really the second step. And so remember, let me actually go to the next slide. Remember that ATP and NADPH were both made during the light-dependent reactions. Remember where they were made, right? So the photosystems 2 and 1, and the ATP synthase, and the proteins involved in the electron transport chain, those were all in the thylakoid membrane, and the NADPH and the ATP were all made in the stroma, okay? And so, again, to take you back to where we are, where photosynthesis takes place, we're in a leaf, right? That's going to be the main organ of photosynthesis. And between the epidermis, between the upper and lower epidermis, this central region will be called the mesophyll. And in here we will have palisade and spongy, or just parenchyma cells, depending on what type of leaf we're looking at. But in any case, inside these cells, we have chloroplasts. And so that's going to be the site of photosynthesis. So all these green little oval to spherical structures are all chloroplasts, okay? And so remember again, in the chloroplasts, the light reactions, right, the light-dependent reactions take place first. And again, those produce ATP and NADPH. And then following that, we will have the light-independent reactions. And so the light-independent reactions are also known as the Calvin cycle. And so that's why at the top of this slide, you see Calvin cycle. These reactions used to be called the dark reactions. And, you know, it was decided to change that name, which is great, because sometimes students would think that the dark reactions only happen in the dark, and that's just not true. So the Calvin cycle or the light-independent reactions can take place during daylight or at night. They don't require light. Okay, that's the key thing. The first part of photosynthesis, the light-dependent reactions, of course, do require light. And so the only way that cells are generating ATP or NADPH to be used in the Calvin cycle is if light is present. Okay, so the Calvin cycle is another term that we use for the light-independent reactions. And these reactions are going to take place in the stroma of the chloroplast. So we're still in the chloroplast. Very importantly, one of the reactant molecules needed for the Calvin cycle is carbon dioxide. There has to be a constant supply of carbon dioxide from the atmosphere in order to drive this process. And so we mentioned this before, but just as a reminder, in the epidermis, and this picture just on the lower epidermis, but in some plants it'll be on the upper epidermis as well, there will be these pores, which are also called stomata. And these pores, these openings, are going to allow for gas exchange. So CO2 will come in and O2 will go out. And remember again that these stomates can be opened and closed according to temperature and the amount of humidity in the air. Okay, so there's different factors that can lead to these being opened or closed. When they're open, gas exchange is allowed. And so remember again, CO2 comes in and it's going to make its way, you know, to all of these cells. It gets taken up into the cell and it's going to diffuse into the stroma of the chloroplast. And that's where the CO2 gets used. Okay, and again, I want to just hit this home one more time. There needs to be a constant supply of CO2 in order to drive the Calvin cycle. Without it, photosynthesis doesn't work. Okay. So here again is just a picture of a chloroplast, just to remind you. And so again, these are going to be found inside of mesophyll cells in the leaf. Okay, and so we've got different spaces. So remember that in the thylakoid membrane is where all of the components for the light-dependent reactions are found. And then out here in the fluid, kind of surrounding these thylakoids, is called the stroma. And so the NADPH and the ATP made during the first step are in here, right? So they're going to be in the stroma, and those are going to be used in the light-independent reactions, or the Calvin cycle. In the Calvin cycle, there are 3 stages. And so we are going to talk about the fixation stage. Okay, and when we talk about fixation, we're talking about carbon fixation. We'll then talk about the reduction phase. And lastly, we'll talk about regeneration. So there's 3 stages or phases that are part of the Calvin cycle, or part of the light-independent reactions. Okay, so let me move my camera here. And so we're going to go through these steps really just briefly. So stage 1, okay, again, we're in the stroma of the chloroplast. And this is what happens during stage 1. So the reactant molecules, where's my pointer, include carbon dioxide. So up here at the top of this Calvin cycle, we've got an input of 3 carbon dioxides, okay, entering 1 per cycle. Okay, so in order to get this thing to drive, right, we have to have 3 carbon dioxides. We also need ribulose bisphosphate, which for short is just called RUBP. And so that is shown right here. We need 3 of these, so you can see 3. And RUBP has 1, 2, 3, 4, 5 carbons, so it's a 5 carbon with 2 phosphate groups on it. Okay, CO2 is obviously just 1 carbon. You have 3 of them, so you have 3 carbon compounds. And the last thing you need to start this process is an enzyme called Rubisco. And so that's right here. Rubisco is a really cool enzyme. It's the most abundant enzyme on the planet. It is required for photosynthesis. Okay. Okay, so what happens is Rubisco, this enzyme here, catalyzes a reaction in which these carbon dioxide molecules react with RUBP, right, ribulose bisphosphate, and we end up with 3-phosphoglycerate, which can also be shown in shorthand by 3-PGA. Okay, so carbon dioxide, again, comes in through the stomates. It's from the atmosphere, and carbon dioxide is an inorganic molecule. During this initial step, when it combines with RUBP, we form an organic molecule, again called 3-phosphoglycerate. Okay, and from the 3 carbon dioxides and the 3 RUBPs, we end up with 6 of these 3-phosphoglycerate molecules. So that's carbon fixation. That's the first step. Slide. Still the same figure. Okay, so we just ended right here with this molecule. Okay, so carbon has been fixed. So carbon from the atmosphere has been come in and it's been incorporated into this molecule. So what happens next is step 2. And in step 2, this is called the reduction phase. And so this is where ATP and NADPH, both produced in the light reactions, they're going to be used to reduce the 3-phosphoglycerate to G3P. Okay, so let's look at that. So here are the 6 molecules of 3-PGA, 3-phosphoglycerate. Here's our ATP, so 6 of them, okay, 1 per 3-PGA, get used, okay? And so we end up with 6 ADP molecules, adenosine diphosphate, and we end up with this molecule right here, which is called 1,3-bisphosphoglycerate. So it looks just like this molecule, except for it's got a second phosphate group on it. Where'd that phosphate come from? ATP, right? We know that that's how ATP works. It donates its terminal phosphate. Okay, so that's what just happened here. So again, we have 6 of these 1,3-bisphosphoglycerate molecules. What happens next is NADPH comes in. And NADPH is going to give up its electrons. And so these molecules of 1,3-bisphosphoglycerate, they get reduced and NADPH gets oxidized. And as that happens, the inorganic phosphate group right here also gets pulled back off. Okay, and during that process, we form 6 molecules of G3P. Okay, now notice the ADP, the PI, the inorganic phosphate, and the NADP plus, they're all going to get recycled, right? They go back and they're going to be used in the light reaction. Okay, so again, just like rechargeable batteries, right? ATP is like a rechargeable battery. We can pull that terminal phosphate off. That's a transfer of energy. And then we can put that phosphate back on. Now it gained energy. The same thing with NADPH. We can put electrons on it. That's going to, that represents energy. We can pull electrons off. Okay, so those are molecules that get recycled. Okay, so that's the reduction phase. Let me just add to this just a little bit. So the 6 molecules of G3P, which again stands for, do I have it written anywhere? I don't know, glycerol 3-phosphate. So these 6 molecules, one of them is an output, is a product. And so one of them will eventually become glucose or other organic molecules. The other 5 are actually going to be used in phase 3, which is called the regeneration of ribulose bisphosphate, or RUBP. Okay, so just like in cellular respiration, when we looked at the Krebs cycle or the citric acid cycle, we had to recycle oxaloacetate. The same thing here, we have to recycle ribulose bisphosphate, RUBP, so that there's RUBP ready for the next cycle. Okay, so not all 6 of these become organic molecules. Instead, only 1 does, and the other 5 are used to regenerate RUBP. Okay, and so this does require energy, okay, because we're going to build a 5-carbon molecule, 3 of them, from 5 3-carbon molecules. So we got to put a couple of these together. Okay, so energy is used. So ATP comes in. It's going to transfer its phosphates, and we end up with 3 molecules of ribulose bisphosphate, RUBP, and that's our starting material. So we've regenerated our starting material, which is great. Notice here the ATP that is used. We still have 3 ADP, right, remaining. That's the product. And those will go on just like a rechargeable battery. Okay. The light reactions will produce a proton gradient, which drives the ATP synthase, which will put ADP and inorganic phosphate together to form ATP again. Okay, so your cells are really smart and efficient, so they recycle materials. So here is a summary slide of photosynthesis in total. Okay, so remember, we got photosystem 2 and photosystem 1. Photosystem 2 is responsible essentially for generating a proton gradient, which drives the synthesis of ATP. Photosystem 1 is responsible for, again, producing high energy electrons that eventually get handed over to NADP to form NADPH. And so both ATP and NADPH are products of the light reaction. Okay, and again, that's going to involve the thylakoid membrane and the thylakoid space. And our products, the ATP and NADPH, are going to be made in the stroma, which is nice because that's where the next step happens. Remember that photosystems 2 and 1 rely on light, right? So that's the energy source. That's what gets converted eventually into ATP and NADPH. Also remember that photosystem 2 relies on water for electrons. That's where water comes into play. Okay, so our reactant molecules, right, are water, which comes into play during the light reactions, light, which obviously work in the light reactions, and lastly, carbon dioxide. And so the NADPH and the ATP made during the light reactions, they're going to be used in the Calvin cycle along with carbon dioxide to eventually produce organic molecules like glucose, so sugar. Okay, and so again, our 3 reactant molecules are water, light, and carbon dioxide. Our products shown here are oxygen, sugar, so that would be glucose, okay? And some products that, we don't talk about in our general formula, we do regenerate RUBP. We also have NADP plus and ADP, right? Those are still considered to be, intermediate products. Okay, so by the end of this chapter, my hope for you is that you can draw this, and I'll be asking you to draw it, in lecture. You should be able to, explain and draw all the steps of these processes. Okay, so that is it for chapter 8.